according to the previous lecture, we have now a substance also known as hereditary material. And this hereditary material, sorry, I'm not sitting on the proper angle. Hereditary material is also known as the DNA. The DNA is deoxyribonucleic acid, and we have been studying the structure, whole structure of the DNA in a bit more detail. But we have now come to know that there are two strands of the DNA, and that DNA is actually holded. The both strands of the DNA are being holded by the help of the hydrogen bonds, which are actually present between the nitrogenous bases present on the nucleotides. What are nucleotides? Monomers of the DNA, and the nitrogenous bases are present on the are the part of the nucleotides. So adenine is a nitrogenous base, and what is a nucleotide? Nucleotide is made by a sugar, a phosphate group, and a nucleotide, and a nitrogenous base. The hydrogen bonding would only exist between the nitrogenous bases of two opposite part of the strands. For example, if there is adenine, then there would be the hydrogen bonding between the adenine and thymine. It would be like this. ये सारी चीजें जो हैं वो we did study in the previous lecture. But why the DNA is so much important? This is another question. That if we are studying this if this is present in our cell, and more specifically, the DNA is not only present in our cell, it is present in the most important part of our cell, that is nucleus. So why is it so much important? What is the importance of DNA? Can you share anything with me?
Yeah. So, as we can see in our surroundings, that like so many animals, they adapt to their surroundings, and usually they have lots of characteristics and features that are passed on from their ancestors, etc. So, most of them come from the DNA, and if the DNA can be altered, for example, so it may create some other features that help the organism survive. You know, like survival of the fittest, something of that sort.
Okay, you are completely right that if we look in our surroundings, we come to know that if we consider parent 1 and parent 2, and they both reproduce, and whenever they both reproduce, they produce a child, or in human language, in common language, we call that organism as a child, or in biological language we call that organism as an offspring. Whenever the parents create an offspring, they reproduce and they create an offspring, we there is a similarity between the parents and the offspring. There are always a similarity. I'm not saying that they are 100% identical, but there is a similarity between the offsprings and the parents. Where did this similarity come from?
It was passed
Sorry.
on from their DNA.
Yes, where did this similarity come from? It was passed from the DNA. It was passed from the DNA. So if it was passed from the DNA from the parents to offsprings, then there must be something very unique about the DNA. Why not carbohydrate? Why not proteins? Why not fats? Why only the DNA? This is a very brainstorming question, that what is so specific, what is so unique about the DNA, that only and only the DNA contains the information that would be bringing the characters or taking the characters right from the parents to the offsprings. So what is so unique about this? This is the question right now, which I created a few minutes ago, that why is it so much important? It is important because it has specific nucleotides which are also known in the language of genetics. These nucleotides are also known as codes. These are codes. For example, you are living in Abu Dhabi, and you can read Urdu, you can read English, you may read Arabic, you may read you may speak Arabic. But there are certain languages, for example, Chinese, for example, some Persians, etc. If if someone give us a letter which is written in the Persian language, we have to decode that letter into the language most convenient to us. For example, let me put an example according to you. For example, the most convenient language, for example, for you is English. If something comes to you that is written in Arabic, what would be your desire if it is interesting? आपकी ये ख्वाहिश होगी that this novel or this book or this article should be written in English. Similarly, there are certain codes present on the DNA in the form of nucleotides, which are an indication, which is a language. It is actually the nucleotide, according to scientists, it is a language of characters which are going to be defined in the next offsprings, in the upcoming offsprings, in the form of something. For example, if these are codes, DNA contains the codes. Language of this is the language of characters, and these codes must be decoded. These codes must be translated. Codes must be converted into a language that should be understood by our body. I think you want to ask something.
No, no, I'm clear about it.
Okay, so what should be the language of our body, and what are the codes? What kind of codes present on the DNA that must be converted into the language suitable for our own body? For example, um, let's take an example of a group of nucleotides. For example, A, G, C, T, G, C, A, T, G, C, A, G, A, C. 1, 2, 3, 4, 5, 6, 7, 8, 9, 11, 12. Let's put T here. So, whenever we uh, examine the DNA, we come to know that nucleotides are the alphabets of the DNA. These are the alphabets of the DNA. And you know very well that alphabets combine to create a word. So it definitely means that if nucleotides are alphabets, they are creating a words. According to scientists, that the words created by the nucleotides are called as genetic codes. These are called as genetic codes, or I should be calling it words. So it means that the language of the DNA, for example, or you may say that the language of the genes or the language of the DNA is actually the nucleotides. Alphabets are nucleotides, and the words created by the nucleotides are also known as genetic code. Now keep this word in mind, genetic code. It would help you in further concept. So if the nucleotides are alphabets, so it means they are creating many words. For example, the nucleotides sorry, first you have to understand another rule about the genetic code. The genetic codes are always made by, I should be mentioning words here in bracket. Genetic codes or words are always made by the group of at least three nucleotides. Three nucleotides or alphabets. For example, let's take another example. We have almost 26, I think, alphabets in English. 26 or
Yes.
28, I think. So we often say that if we want to create a word, then there must be a combination of two alphabets. For example, if I want to write two, then there must be two alphabets. This is no word, this is no word, this is no word, but this is a word, combination of two words, meaningful combination. For example, 2 TWO2, this is another meaningful combination of the alphabets. And then there is the combination of 5 words. This is the meaningful combination. So it means that alphabets are meant to combine into a meaningful word that should be understood by the by, if we talk about the English, then it must be understood by us. अगर अल्फाबेट्स ऐसे लैंग्वेज में कंबाइन हो रहे हैं ऐसे कॉम्बिनेशंस में जो हम अंडरस्टैंड कर रहे हैं, बट according to genes, according to DNA, according to molecular biology, the genetic code or the words are only completed when these are made by the combination or of at least 3 nucleotides. For example, AG is not a word, CT is not a word, G is not a word, but this is a word. Word 1. And this is also known as genetic
Cool.
code. Similarly, AGC, then TGC is another word, genetic code. Then ATG is another code, and then ACT is another code. These are the genetic codes present. This is the DNA language present on the DNA, which is brought to the offsprings from the parents. वो कैसे आते हैं, वो आपने सेल डिवीजन पढ़ी हुई है, क्रोमोसोम्स की आगे जो है वो आपने ट्रांसफर पढ़ी हुई है. दैट इज द वे ऑफ ट्रांसफरिंग दीज कोड्स टू द नेक्स्ट जनरेशन. अब यहां से क्या होगा, इट वुड बी डिकोडेड, it would be decoded into another language. Okay, what is that language? You know,
Sorry, sir.
what should be that language? Nucleotides are is a language of DNA, but what should be the language in which these nucleotides should be converted? What would be that language? अगर मैं इस न्यूक्लियोटाइड की लैंग्वेज को, इस न्यूक्लियोटाइड की ज़बान को कन्वर्ट करूं, तो कौन सी वो ज़बान है जिसमें ये कन्वर्ट होंगे, कौन सी वो सिचुएशन है? कीप दिस इन माइंड दैट डीएनए always converts its information into the form of proteins, into the form of proteins. जब भी डीएनए से इंफॉर्मेशन डीकोड होगी, जब भी डीएनए से इंफॉर्मेशन नेक्स्ट जनरेशन कोई काम करना होगा, फॉर एग्जांपल एक पर्सन है, उसके अंदर जो है वो, फॉर एग्जांपल देयर इज अ पर्सन व्हिच हैज द ब्लड ग्रुप ए पॉजिटिव. ए पॉजिटिव is actually defined by a protein, and protein is made by the DNA, and DNA is given to us by our parents. So it means the parents, if the parents give us the DNA, then that DNA would define our body by creating suitable proteins. By creating suitable proteins. For example, let's take an example of a cell. This is a cell, and this is a nucleus of a cell. There is DNA strand is present. The DNA would be transferring. DNA would not be destroyed. If the DNA is decoded, then decoding means decoding. It does not mean that if a DNA is converting into the protein, then it would be destroyed and re-established into a protein. No, DNA would remain on its place. It would just be decoded from the language of nucleotides to the language of proteins. Okay, I'm continuously using the word language. I would be very clear about it a few minutes later, but first we have to understand it completely. For example, this is the DNA. We want to create an enzyme. You know about the enzymes?
Yes.
Okay.
There are
What
biological
do you know about
catalysts.
the chemical property of enzyme? What is chemical structure?
Um,
Chemical structure of enzymes are generally, if we call it general structure of enzyme, is a protein. Does that ring a bell? That must ring a bell. that enzymes are made from
Proteins.
proteins, and the proteins are made from the DNA. Which means if we have some enzymes, those enzymes are by the blessing of the DNA. DNA has been converted into the enzymes. So it means the DNA would be converted into the protein, and that protein would be of two types. For example, it can be an enzymatic protein. हो सकता है उसको हम different काम दे दें, हम उसको कहें कि वो enzyme बना ले, ठीक है? Then it also can be converted into another working protein. For example, it can be converted into our muscular protein. It can be used to make our muscles. It can be used to make our hemoglobin. It can be used to make any structure of protein in our body. Any structure of protein, muscle, I have mentioned the muscle. It can create the heart. It can create the connection between the muscles. It can create the antibodies. You must have an idea of antibody. It can create the antibodies, right? So
Yes.
DNA would create a protein and then would specify that what should be the working of this nascent or newborn protein. This would also be defined by the DNA. If the protein is meant to be an enzyme, then it would never be converted into the hemoglobin. It would always be converted into the enzyme. But it is, if it is meant to be converted into the hemoglobin, it would never be converted into the muscle protein or heart protein or antibodies. No, hemoglobin protein is hemoglobin. It would never be an enzyme. So it means that DNA has the property of creating, DNA creates specific proteins. Are you understanding all this?
Yes, sir.
Okay. Now, यहाँ से थोड़ा सा तो idea हमें हुआ that there is a specific thing which is known as nucleotides, which is also the language of characters that has been sent to us by our parents, and that language would be decoded, would be converted into the form of suitable language for our body, and that suitable language is the proteins. Suitable language is the proteins. अगर हम अपनी बॉडी में से वाटर रिमूव कर दें, dry mass, just consider the dry mass of our body. The dry mass of our body consists of 60% of proteins, more than 60% of proteins, which means that the remaining percentage is of DNA, is of RNA, is of carbohydrates, is of fats, is of some other substances. But the 60% part of our body is proteins. That creates the protein very much important for our body. That is why we have to keep eating the proteins so that our DNA can make some many other proteins. You must know that all those gymmers, all those boys and girls which are building their body up to a specific you may say that specific figure, they all are taking the protein supplement. They all are taking the protein supplements. Why? Because whenever they perform the exercise, their protein firstly breaks down. जब भी आप एक्सरसाइज करते हैं प्रोटीन ब्रेक होती है, बड़ी जबरदस्त ब्रेक डाउन होती है. But after some time, they eat the eggs, they take the protein supplements, they take the amino acid supplements, etc., etc. They take the supplements, and when they take the supplements, the DNA captures those supplements, build stronger proteins, and store those proteins into the muscles. Store those proteins into the muscles. So it means that if we want to break our muscles, or if we want to resynthesize, if we want to gain weight, if we want to gain some muscle, then we have to eat the protein so that our DNA assemble the protein into our DNA, sorry, into our muscles, right? So There must be something special, and that special is the nucleotide, the language, the proteins. But there is another thing. DNA, we have DNA. And then I have wrote in this sentence that DNA creates specific proteins. DNA भी समझ आता है. It synthesizes or it creates. Okay, understood. Proteins. Okay, understood. But what is this specific? How the DNA makes the specific proteins? How am I, first answer my question, answer my this question, that how many DNA segments, or how many DNA small DNAs we have in our nucleus?
Sir, could you like repeat the question again?
Sure, sure, sure. How many DNA molecules are present in our nucleus?
Um,
How many chromosomes?
yes, 23.
Is it?
Yeah, yeah.
46? We have
Yes.
46 chromosomes. Similarly, if these chromosomes are disassembled, if these chromosomes are unfolded, then the chromosomes would be converted back into the DNA. It means if we have 46 chromosome, then we have 46 DNA. Then we have 46 small molecules of DNA inside our nucleus. Have I made this point clear, or there is something to clear it?
Yeah, it's clear.
Okay, it means that we have 46 DNA molecules in our nucleus. But if we have 46 DNA molecules, so, does it mean that we create only 46 proteins? If a single DNA molecule is meant to create a single protein, single specific, focus on the word specific. If a single DNA molecule, बेटा हमने आज क्लास 8 बजे स्टार्ट कर दी है.
Yes, sir.
और आपके टाइम के लिहाज से 7 बजे स्टार्ट की है.
नहीं सर, अभी 8 बजे हैं. It's 8:32 at the moment.
8:32 at the moment. uh, it's my time 8:33 right now. What about your time?
So it's 8:33 p.m. over here now. I
My
think
time
it's
is wrong here.
yeah.
2 दिन पहले विंडोज इंस्टॉल हुई, टाइम गलत हुआ. ओके, नो प्रॉब्लम. what was I talking about? Sorry, मैं दरमियान में इंटरप्ट कर लिया, इसको दोबारा रिपीट कर लेते हैं. I was making the point that if we have 46 DNA molecules, then does it mean that if a single molecule is specified for single protein, for example, DNA number 1 is dedicated for a single protein, then it means that 46 DNA molecules are dedicated for 46 proteins. Then it is not an understandable point. We have thousands of characters, but how come only 46 DNA molecules, thousands of proteins? Even only if you examine the antibodies, the variety of antibodies in our body, these are more than 46. If you examine the characters in our body, these are 1,000 times more than the 46, the figure of 46. And if a single DNA synthesizes single protein, then it would not fulfill our demand of proteins. It means there must be something else. मैं जो पॉइंट बनाना चाह रहा हूँ वो पॉइंट अब मैं लिखने लगा हूँ. उसको हम हाइपोथेसाइज करेंगे, उसके बाद उसको अंडरस्टैंड करेंगे. देयर मस्ट बी समथिंग एल्स. मस्ट बी multiple proteins are synthesized. Multiple proteins are synthesized by a single DNA. ये पॉइंट कुछ समझ आता है. Why? If a single DNA synthesizes multiple proteins, then 46 DNA molecules can combine to create a huge amount of proteins. For example, if a single DNA, one DNA can create 50 proteins, then you can assume how many DNA molecules we have. 46 DNA molecules we have. So 46 DNA molecules can create, I think, you can easily calculate 46 into 50. It is an
Yeah.
you may say that considered figure, supposed figure, but ये point कुछ valid लगता है कि single DNA molecule creates multiple proteins. But एक और
That's
question
true.
arise होता है, how? एक DNA molecule इतने different proteins कैसे बना सकता है? So let's come to the answer. If you examine a DNA molecule, then you come to know that a single DNA molecule is functionally divided into some segments of variable size. Segments of variable size. This segment create protein 1. For example, this segment create protein 2, this segment create protein 3, this segment would create protein 4, 5 and 6, which means that every DNA is functionally divided into multiple segments of variable length. Multiple segments of variable lengths. Of variable lengths. You know what should we call those segments?
Um,
Those segments are called as genes. Multiple segments of variable lengths. So it means that this would be converting into different protein. This segment would be decoded into the different proteins. This would be decoded into the different proteins. This would be decoded into the different proteins. All of these segments would combinedly be called as the genes. So if we come back to our point that in our nucleus we have 46 DNA molecules. All those 46 DNA molecules are functionally divided, not physically. Keep this in mind. These are not physically divided. ये सारे आपस में जुड़े हुए हैं, फिजिकली जुड़े हुए हैं, फिजिकली डिवाइडेड नहीं है, बट दीज आर फंक्शनली डिवाइडेड into multiple segments of variable lengths, which are decoded into proteins, and these functional division or functionally divided segments are known as genes. Have I made the point?
yes, sir.
Okay. Now we have understood the genes. So another thing, how the gene create the protein. Another point. सवाल ये है ना कि जीन जो है उसकी तो लैंग्वेज है न्यूक्लियोटाइड, एंड द लैंग्वेज ऑफ प्रोटीन, व्हाट इज द लैंग्वेज ऑफ प्रोटीन? व्हाट आर द अल्फाबेट्स ऑफ प्रोटीन्स? और लेट मी मेक इट इजियर फॉर यू, व्हाट आर द मोनोमर्स ऑफ प्रोटीन्स?
Amino acids.
Yes. So it means the language of the proteins are amino acids, and the language of the genes or the language of DNA is nucleotide, and this would be converted into this. But another question is standing here, how? We have created many hows. Since the start of the lecture, we have created many hows. So we are one by one answering our hows. So the nucleotide would be converted into amino acids, but why, how would it happen? Let me check something. Okay, some things are still here to be cleared. Now, first of all, if we call that, if we say that DNA would be converted into the proteins, then there are some several steps which involve this whole phenomena. What are those steps? First of all, the DNA would be converting into a specific single molecule. You know very well that DNA is a double-stranded molecule. First of all, the DNA would be converted into another molecule. Let me be very specific. Let me tell you a single, each and every step about this. Keep this in mind that the DNA does not exit the nucleus. Let me tell you one thing, another thing. Proteins are synthesized or created outside the nucleus. Proteins are synthesized outside the nucleus. And DNA does not exit the nucleus. So how come that the protein would be synthesized outside the nucleus and the DNA would never leave the nucleus? How is it possible? This is the question I'm going to answer. That let me write it here. If the DNA does not exit, and protein is made outside the nucleus, how the how this phenomena is possible. Now let's make this phenomena possible, because it is possible. The problem is this. The problem is not that it is impossible. The problem is it is possible. How would it be possible? The DNA first would be converted into a movable molecule, a molecule that would be moving and that can leave the nucleus. That molecule would be known as RNA. More specifically, that molecule will be known as messenger RNA or mRNA. इसको हम डीएनए को बजाय बाहर भेजने के we would be converting that DNA into RNA so that RNA can move outside the nucleus and take the information from DNA to outside the nucleus so that the protein can be synthesized. इसलिए हमें messenger RNA बनाना पड़ेगा so that the DNA does not have to leave the nucleus. So first of all, the DNA would be converted into the messenger RNA. This phenomena would be known as transcription. This phenomena would be known as transcription. Let me show you here transcription. Transcription. The transcription is when we open the DNA. Look at this diagram. We open the DNA. We create. Do you have the book in front of you right now?
Yes,
Are the
sir.
diagrams are like this?
Yes, sir.
Okay. Uh, we open the DNA. We copy the DNA in the form of messenger RNA, which is actually single-stranded. Now it is single-stranded. This messenger RNA would be sent outside the nucleus to create the proteins. Create the proteins. There is another thing I would be telling you. Key point here. Okay, I would be telling you, but let me make it clear. So the messenger RNA would come outside the nucleus. Where would it come? The location now matters. It either can come into the cytoplasm or it can reach into a very specific part of the cell which is known as endoplasmic reticulum. You know about this?
No.
Okay, let me give you a very small idea. If this is a cell and this is a nucleus, there is a huge network of small membranes outside the nucleus. Just like you fold the clothes and put those clothes on each other, create a stack of clothes, then it would be like this. This is a nucleus, and this portion of folded clothes is known as endoplasmic reticulum. So either the messenger RNA would be sent to the cytoplasm or it would be sent to the endoplasmic reticulum. Now here it would be converted into the proteins, and this conversion would be known as translation. Focus on the point, when we converted the DNA into messenger RNA, it was called as transcription. And when we converted the messenger RNA, now the messenger RNA has been sent to the cytoplasm. If we send the messenger RNA into the cytoplasm and the messenger RNA now would be converted into the proteins, then this phenomena would be known as translation. Let me make it very simple. DNA to messenger RNA. This phenomena is known as transcription, and the messenger RNA into proteins. This phenomena would be known as
Translation.
translation. Translation, and now it would be converted into the now the how it would be converted. We have to take a very slight concept or a glimpse of how the translation occurs or how the protein is made. Uh, Let me be very clear about this, because in the previous actually there is a small confusion here in the syllabi. If you focus on the syllabus of those students which are going to appear in 2021, those students have entirely different syllabus, entirely different curriculum, many things. If I look upon the books, then I told you earlier in the lecture that your major subject, major portion of your curriculum is taken from the IGCSE syllabus, IGCSE curriculum. Major portion of this right now your curriculum is taken from there, and that the curriculum that is for the previous class, or you may say who are going to appear in the in 2021, the curriculum for them is entirely different. So I have to make it very sure that either you should be reading the topic or not, you should be studying the topic or not. At this point I have made clear cell functions by controlling the production of proteins including enzymes. State that the sequence of bases in a gene determines the sequence of amino acids needed to make a specific protein. This is we also have discussed this. Understand that different sequences of amino acids give different. According to curriculum, we are not meant to uh, have the detail of this. Uh, this detail. We are not meant to study this detail. Thank God. This is because there is nothing mentioned about the messenger RNA, nothing mentioned about this transfer RNA. This is transfer RNA. Nothing mentioned here, and Alhamdulillah, this is no. We are not meant to study this one. Genetic code is not mentioned here. So the actual use of genetic code was in this topic. What I told you earlier was just an understanding. The actual use was here, and we are not meant to study this, so we should not worry. Okay, डीएनए ने मैसेंजर आरएनए बना लिया, मैसेंजर आरएनए ने प्रोटीन्स बना ली. Okay, now proteins, how the proteins are different. How the proteins are different. I think you can answer this on your by yourself. Yes, Zainab, can you answer this?
Um, so proteins are made up of monomer molecules called amino acids. So maybe maybe it could be that they're using maybe like different proteins have different sorts of amino acids and in different amounts.
Okay, if different proteins have different amino acids, then who actually is responsible for the sequence of amino acids in the proteins? Isn't it the DNA?
Yes, because the DNA is making the protein. So
Yes, 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 yes. It means that if I am making a protein 1 and protein 2, then it means that uh, if I have the DNA sequence AGCT, 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 okay, then I have the sequence TCGA, TCGA, TCGA. Now, if I have this sequence of AGCT, AGCT, AGCT here, and the group of three is defining the amino acid, then this is amino acid 1, this is amino acid 2, this is amino acid 3, and this is amino acid 4. These all are synthesized, or these all are determined by this sequence of DNA. This is the DNA and this is protein. Now when you come to the next protein, the genetic code now has become entirely different comparative to this one. The genetic code has now entirely different. It means that the amino acids of this protein would be different than the amino acids of the protein 1. That is why two enzymes differ between each other. For example, let me give you an example, which is a very good example. You have studied in the previous class about the amylase and pepsin.
yes,
What is common feature between them? Both are
both are proteins.
indirectly, you are right, both are enzymes. If both are enzymes, then what is different between them? Both are enzymes, both are biologically active, both are reducing the demand of what we call activation energy. I think you can remember from the previous class, the demand of activation energy is reduced by the help of enzymes. So both are known by the same name, enzyme. What is different between them? Their structure.
Genetic, yeah.
Why? Yes, genetic code indirectly, you are right. If you a genetic sequence or genetic code, if you reverse the sequence, for example, protein, messenger RNA, and DNA, if you reverse this sequence, then you come to know then all that always, if two proteins are different, only and only DNA is responsible for the change in the sequence of two proteins. ओनली एंड ओनली द डीएनए रिस्पॉन्सिबल. लेकिन हम ये बात भी कर चुके हैं कि जब डीएनए चेंज होगा, then the amino acid sequence would also be changed. So it means the amino acid variety of amylase and the amino acid variety of pepsin is changed. So the two proteins are always changed if their source DNA is changed. For example, you know about thalassemia? I
I've
think you
heard.
should. Yes, it is a very dangerous disease in which hemoglobin is not made. Hemoglobin is not made, and these persons have continuously almost 4 days a week they have to go to the blood bank, and they have to take the they have to you may say they have to be donated the blood. The donors regularly come to those blood banks, and they donate their blood as a sadqa for those persons, and these persons cannot live without continuous donation of blood. Thalassaemic patients, these are called. So it means that if we have the hemoglobin, we have hemoglobin, and we are non-thalassemic. A normal person, there is a normal person, and there is a thalassemic person. So normal person has normal hemoglobin. Now I am talking about the same protein. Focus on the point. I am talking about the hemoglobin in both cases. What is happening? In normal person, the DNA sequence responsible to create the hemoglobin is normal, is right, is working properly to create the hemoglobin. But in thalassaemic patient, all that is responsible is DNA, or you may say gene, gene of hemoglobin. This is a small example which I found necessary to mention here, that gene of hemoglobin is actually is responsible for the change in the structure. In this case, hemoglobin is useless. In this case, hemoglobin is useful. So every time responsible is the gene. You must have listened something about the cousin marriage.
Yes.
What happens in the cousin marriage? That sometime if a cousin has the bad genes, then there is a you may say a bigger chance of female or male cousin to have that same defective gene. Bigger chance. I'm not saying that it would always occur, but there is a bigger chance. So that their child would suffer in the life later. मेरा एक दोस्त है, वो शादी नहीं करता, उसको वो हीमोफिलिक है, वो ये भी एक जेनेटिक डिजीज है. The gene is abnormal for the blood, so he does not marry. कहता जी, why should I make the upcoming generation suffer due to my genes? It's a huge statement to make, but he is huge by himself. His personality is huge. He's a very great person. Anyways, I have to be very, very specific. That is why I keep reading the curriculum. This is the point. State the mutation, meiosis, random mating, and random fertilization mutation. And here's mutation. For the next day, you must remember that there is something known as genetic code, which is a group of how much nucleotides?
3.
Very good, well done. 3 nucleotides, and that genetic code is converted into another language known as amino acid language. So if I have a word TWO and someone gives me an article which has TWW, would I able to translate this word on my own until I was told? Can I translate this word on my own? I don't think so.
No.
For example, there is another word, twice. Someone writes is TWECE. What is this? I cannot translate on my own. Same thing happens in Mutation. If something is changing, that is mutation. That can be good or that can be bad. We will be discussing in detail, inshallah, tomorrow. Anything to ask? Any problem?
No, sir.
Anything to share? Anything you want to improve in the lecture? This is the most important point.
Sir, everything's clear, alhamdulillah.